In this video, we will discuss how the Siglec receptor can be act as a new immune checkpoint inhibitor in the case of cancerous cells. And why I am talking specifically about cancerous cells is because cancerous cells has this overexpression of the sialic acids that can be seen here in this figure. And this is this figure on the left hand side is the comparison between the checkpoint inhibitor that has been identified as one of the checkpoint inhibitor that has been identified as PDL1, and they compared how this checkpoint inhibitor function is correlated with this Siglec receptor inhibitor function. So in the case of checkpoint inhibitor function, what happens is that this PDL1 that is expressed on the tumor cells interact with this PD1 receptors on the immune cells and then trigger and the phosphorylation that will leads to the deactivation of the T cells. And similar kind of things happens when the silated proteins or lipids interact with the Siglec that are expressed on the immune cells that will lead to the phosphorylation uh, of the Siglec receptors in the within the cytosol of the immune cells, and that will further deactivate the T cells. So this is in general how the Siglec receptors can be act as a new checkpoint inhibitor uh, that is similar, whose function is similar to this PDL1 and PD1 interaction. And now, what are the different kind of Siglec receptors expressed on different immune cells uh, act uh, act as a checkpoint inhibitors are expressed are mentioned in this figure on the left figure on the right hand side. So, and these interactions are usually occurs in the tumor microenvironment, as the solid tumors has the property to develop tumor microenvironment that will sustain or that helps the tumor cells for its proliferation, and at the same time helps in the immune evasion. So one of the way by which immune evasion occurs is via this Siglec interaction, via these sialic acids expressed on the tumor cells interact with the Siglec receptors expressed on the immune cells. And so in the case of these T cells, how the suppression of T cells occurs by the tumor cells is similar to that I have mentioned in the figure on the left hand side. That is the sialic acids that are expressed on the overexpression of the sialic acids on the tumor cells interact with this Siglec 9 receptors that are expressed on the T cells, and that will leads to the that will further leads to the phosphorylation within the cytosol of the T cells, and that will trigger suppression of the T cells. And in the case of this NK cells, what happens is that the NK cells has two kind of Siglec receptors that leads to the deactivation of the NK cells, that is Siglec 7 and Siglec 9. And the Siglec 7 and Siglec 9 interacts with the same overexpression of the sialic acids, as Siglec has a property to interact with the sialic acids. This, in the case of NK cells, it's Siglec 7 and Siglec 9 that interacts with the sialic acids on the overexpression of the sialic acids on the tumor cells, that leads to the deactivation of the NK cells. And this tumor associated macrophages, and what happens in the case of tumor associated macrophages, Siglec receptors interact with the overexpression of the sialic acid in the tumor cells. In this case, uh, this interaction will leads to the polarization of tumor associated macrophages into this M2 phase, and this M2 phase is known as pro-tumorigenic phase, where this tumor associated macrophages will promote will in the M2 form or in the M2 phenotype promote the tumor progression. And this video about this how this tumor associated macrophages helps in the tumor growth and proliferation is already been done in our one of our previous videos. So this is just three examples that are shown here, that is on the T cells, NK cells, and this tumor associated macrophages, how the Siglec acid sialic acid and Siglec receptor interactions leads to the suppression of the immune response. So I hope you got an idea how the Siglec receptors are act as a new immune checkpoint inhibitors. If you need to understand more about this in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.